Today, we will be looking at metagenomics data analysis with OmicsBox. The goals of this presentation are to show you how you can do quality control and assessment with OmicsBox, how you can do taxonomic classification, Kraken 2, how you can we will have a look at the results, and also do some differential abundance analysis of the different taxa that we have been able to find. The data that I chose today comes from a paper from the Nature journal with the name Comprehensive 16S rRNA and Metagenomic Data from the Gut Microbiome of Aging and Rejuvenation Mouse Models. So the gut microbiota is crucial for host health and longevity. This study used various methods, including 16S rRNA and metagenomic sequencing, to analyze changes in gut microbial communities and their functional potential in aging mouse models. The generated data provide valuable insights for developing therapeutic strategies to promote healthy aging. And they uploaded the data under this project name, where they deployed over 100 rRNA 16S samples and over 100 samples that have been sequenced with whole genome shotgun sequencing. There are two big groups here in their experimental design. They have the aging samples, where they sequenced after 1 week, 4 weeks, 20 weeks, 50 weeks, and 100 weeks. Here, 20-week-old mice are considered young, and 100-week-old mice are considered aged mice. The rejuvenation samples are the samples that have been treated somehow with typical rejuvenation techniques. So there is co-housing, where they put young and aged mice into the same cage. We have a parabiosis group with mixed young-aged, young-young, aged-aged. And we have a serum injection group where they transferred from young to young and from young to aged. And we have an AK treatment group where they orally fed Akkermansia bacteria to aged mice, or just the prebiotics for that bacteria. Let's remind us a little bit about the sequencing techniques. So targeted 16S sequencing focuses on specific regions of the rRNA gene in bacteria and archaea. It offers resolution up to the genus level, sometimes species level, and is generally lower in cost due to the targeted sequencing. This method provides information on the composition of microbial communities, and that's about it. Whole genome sequencing, on the other hand, analyzes entire genomes of all organisms in a sample, including bacteria, archaea, viruses, fungi, and eukaryotes. It offers high resolution, identifying down to species and even strain level, but also comes at a much higher cost due to the need for deeper sequencing coverage. This method provides comprehensive data on microbial composition, functional potential, genetic diversity, and interactions. So depending on how you sequence, there's different things you can achieve. And now let's look at what I have prepared today. We want to do some quality control of our short reads. We want to do some taxonomic classification, and we want to explore the results. Let's jump right into it. So this is OmicsBox. This is the software. We have a menu at the top with the different modules We will be looking at metagenomics today. There are a lot of things here. I will show you a few. This is the main visualization or result tab here. I will close those. And then we have on the bottom left, the progress tab where we can see what is currently running, nothing, and what we did in the past. We have a file browser here where we can see files on our disk and open them directly in OmicsBox. And we also have the cloud files here. I will explain something about that later. So let's see. I have a folder here which is called Metagenomics Webinar, and then I have Amplicon and WGS. So I have the raw Amplicon reads here and also the raw WGS reads. So let's do some quality control. Let's see what we have. We can go to General Tools, FastQ Tools, and then Short Read Quality Assessment with FastQC. Put this here a little bit more to the corner. So we select our input reads, and then we provide additional adapter sequences, should we know that there is special adapter sequences in our read data. When we hit the run button, I already did that, I prepared that, and we will get this type of result, which is a table. You oftentimes get a table in OmicsBox, one sample per row, some basic information. Let's look at the first sample. Show a detailed report. So total number of reads, 70,000. Read lengths range from 70 to 251. And we have the different statistics. They have different flags. Sequence quality passed, per sequence quality scores also passed, and per base sequence content failed in this case. Let's see what's going on. Okay, this failed because we would expect a uniform distribution, like a flat line for the different nucleotides over all the sequences. But since we're sequencing the rRNA gene, we expect this bias towards a pattern. So we don't have to act upon that. The GC content is also biased. We would also expect that with 16S data, very conserved regions. And we also have a lot of overrepresented or sequence duplications. So yeah, I mean, we're sequencing a very conserved gene. This is expected. So we don't have to act on that our data. We can consider this okay. Let's look at the results from from analyzing the whole genome shotgun reads. Let's look at the first sample. Okay, so here we have 2.7 million reads. Read length, very long. And like they are all 251. They have not been treated before. And the quality is bad. Okay, it's flagged as failed. What's happening? Okay, the 3' end, there is something. Okay, they really degrade here. The per base sequence content looks a lot better. Yeah, it's a flat line. We have some bias here in the end, and there is something related to primers, I guess, in the beginning. We have some ends toward the end, end nucleotide unknown, and we have something is going on with the adapters. So someone didn't do their job. The adapters have not been removed. We have Nextera adapters. Okay, so we have to get rid of all that because the data is very noisy. So what I can do, I go to FastQ tools again and go for pre-processing with Trimomatic, just a tool that we implemented here. So I select all my FASTQ files, and then I have to tell the software that this is paired end data. In this case, we have paired end reads here because, and this is very important because if you have single end data at single end, that's okay. Every sample or every file is treated one by one. But if you have paired end data, it's very important that you select paired end because then the pairs are treated together. And if there's a read removed from the upstream file or the downstream file, it will also be removed from the other one. So we will end up with the same number of reads or down and upstream, because otherwise the next software you will use on that data will fail or give you wrong results, or the data um, will not be treated right, will not be interpreted right. So make sure to always select paired end if you have paired end data. That's very important. On the next page, we can tell the software to remove adapters. We've seen that we have Nextera adapters, so I can select that here. I can also provide custom adapters should I have them. I can provide a FASTA with custom adapters. We also want to do some trimming from the 3' end with a threshold of 25 quality. We can do more quality trimming, sliding window, adaptive quality trimming. I decided to filter on average quality and also filter by minimum length of 50 to guarantee minimum length of the reads. And then we select the folder where to save the trimmed reads and hit the run button. And after a few minutes, we get the trimmed reads. And then we can run FastQC again to see if we improved. I already did that. Let's look at the first one again. So comparing quality here from failed. I mean, first of all, we can see the number of reads was reduced, or maybe 100,000 reads have been removed. And we also have shorter sequences now. Okay. And then the quality is now good. Let's see. Yeah, that looks much better. Maybe we have even been too um, yeah, conservative here, but yeah, I mean, this is super fine now. This looks very good. The purpose sequence content still fails, but like this line here looks better. And this is still due to the sequencing protocol, I think, and can be ignored. We have the N content has gone away, and the adapter content also has gone away. So we successfully removed the adapters. That's super fine. So we are happy now, and we can go on with the next step of, yeah, I called it quality control. I don't know where to put it, but yeah. So if we're sequencing the whole genome with the whole genome shotgun, we also get reads that belong to the mouse genome because we're sequencing the mouse gut, so we will eventually get mouse, and we don't have mouse in our target database for the taxonomic classification, so we will get reads that will be unclassified or falsely assigned to maybe bacteria, and we don't want that. We want to remove that noise, so let's get rid of those, and this is called contaminant removal. So again, we have paired end data, select paired end, please, and then select your reads and the target genome. With this example, we have mouse here, so I can select mouse. If I have a plant or something that is not like something special, I can provide my own genome here. I can also provide one plant, run this through, select another plant, run this again to subsequently remove all the contaminants that I might have or expect in my data. Okay, you could do that. I only run that once with a mouse genome. I save the contaminant-free sequences to a folder, and you can also select the ones that actually are the contaminants, like the mouse genome reads, if you want them for something, I don't know. I just wanted the clean reads. Hit the run button, and we get a report. We always or usually get reports after running any analysis here in OmicsBox. Basic information about the samples, and then an overview about the alignment, how many sequences have been aligned, mapped, duplication rates, a lot of statistics. I will not go into detail, but this graphic, you also get that as a result, is much more interesting. So we can see that there have been quite some contaminant reads here. So we have up to 28, 25, and 17% in some of the samples. And we only kept the red parts, the ones that didn't map, the ones that are free of mouse genome sequences. And this already like this always happens, although in the wet lab they try to remove the contaminants, that you always get some of them after sequencing and you have to remove them. Okay, so we can move on with taxonomic classification. Kraken 2 and the different databases. Let's talk about that. So in the metagenomics module, we can go to taxonomic classification and select Kraken 2. So Kraken 2 is a highly efficient and accurate taxonomic classification system for metagenomic sequences. It uses a k-mer-based approach to classify sequences by comparing them to a database of known genomes. Kraken 2 can handle large datasets quickly, making it suitable for both 16S rRNA and whole genome shotgun sequencing data. Yeah, its speed and accuracy make it very popular. It's very robust, works very well. Um, we you can only find in your samples what you have in the database. So if I have bacteria in my database, I can find bacteria. But yeah, I will get to this in a moment. So we have different databases here. So this is the standard database. It contains all the ResSec genomes for bacteria, protozoa, archaea, fungi, and virus available on NCBI. It's very big and contains the whole genomes. Then we have some 16S databases here. Silva, Green Genes, Green Genes 2, GTDB, and also something here called WOL2. You will not see that if you open up OmicsBox. I will explain to you in a second why. Mm, so yeah, depending on your data, like if you have 16S data, you should use a 16S database or the RefSec database. And if you have whole genome shotgun data, do not use a 16S database because it is very likely that you will yeah, have very little numbers of classification. You want to use the RefSec database then, or a database that you provide on your own. So that's the next thing. You can also provide databases on your own. You can upload data to your personal cloud file space in our in OmicsBox. So you can drag and drop data here. I created a folder with the name WOL2, which refers to Web of Life. They yeah, provide data for taxonomic classification, and I was playing around with their database to see if it fits. And so you can just put the Kraken 2 related files in there, and then the database will appear in this list, and you can use it. It will only appear for you. Okay, so you upload it, and then you can use it. You can create your own or download any database from the web that is formatted in Kraken 2 format. If you need help with that, just send us an email or look into the manual. It's explained there as well. So yes. And I also said earlier that you can only identify in your reads what you have in the database. So we have an option here, which is called database info. And there you can select any of the databases that you have available. And then it will give you a corona chart. Let's look at one example here. It will look like this. And this is a representation of the whole database. We have nearly 50,000 different taxa in here. Um, viruses, bacteria, archaea, eukaryotes, and you can look around, search for anything. What do I know? SARS. To see if it is available and if you could detect it with your um, with this database when you use it. In this case, we have coronavirus in here, just as an example. Okay, so let's see. We have decided our database. We have the samples that we want to analyze. We select paired end, and then we can do some filtering here. We can, yeah, there's a confidence score assigned to every assignment. Um, we can filter for that, and we can also filter for minimum hit groups assigned to each classification. And then we hit the run button, and depending on the data set, after half an hour or something like that, you will get the results. And they will look like this. I will show you two of them. So we are now at the exploration part. We will now look at our results that come from Kraken 2. So you will get a table once more. Um, with one taxonomy per line. And on the left, we can see the rank. So this is a species, this is a family, this is an order, and we can filter. Maybe I only want to see the genus level here. So everything will be genus. Yeah. And I can also sort. First the domains, then the kingdoms, then the phylums, and in the end, species, and that's it. I can see the taxa IDs and the scientific names. I can also see all the different samples here, like the counts for each sample. I have all the different samples here. And I can see that I have very little unknown and 144,000 reads aligned to bacteria, 0 to virus, 0 to archaea, and so on. Those are cumulative counts, like summed up counts. Like this means that there are so many reads assigned to bacteria or something more specific. Um, if you want the original counts from Kraken, you can export them here on in the side panel on the right. You can export the data as it is, or in Kraken format. So you will see that there are some reads which are aligned to bacteria because Kraken didn't know anything more specific about it. This can also happen at other ranks. But yeah, maybe it's not that important. Um, what options do we have with this data? We can, yeah, sort, filter. We can remove samples. Like if there's an outlier sample, we want to remove it. We can rename samples. We can extract sequences. So maybe we want to look at all the eukaryotes from from my samples. I want to analyze them again or somewhere else. Maybe I want to assemble the data, the reads, or I want to do a gene finding or look at the functional potential or whatever. So I can go here and I could say extract sequences. I provide the FASTQ files I used when analyzing them, and I select the samples from the ones I want to extract the reads belonging to eukaryotes. And I can also inverse the extraction, and then everything that belongs to this will be exported to this folder. This can be very handy. We can also combine um, different results. So maybe I have this result and I have this result, and I want to combine the data together to be able to create statistics on the whole thing. I can click here and add samples from another um, object. Okay, so let's look at some reports. So I analyzed um, this is the result of amplicon with green genes, and this is another report where I analyzed the whole genome shotgun data with the RefSec database. So to give you some different ideas here. So the report. Um, General information about my samples, how much has been classified. Like you can see here, we have 99% or more. So this is ridiculously good for the amplicon data set. With the whole genome sequencing, um, we only have up to 28%, 36. So numbers are much lower, more noise. Um, yeah, whatever. We don't know. Um, we get some alpha diversity at species level statistics here. The Shannon index and the normalized Shannon index and the inverse Simpson index. So the higher those numbers are, the more diverse are those samples, and they both are like the Simpson index is influenced more by most abundant species, and the Shannon index is more sensitive to rare species. But yeah, you can read up on that to see what they tell you exactly and how to interpret them. And then we have the percentage of classification per rank. At the domain level, as we have seen, 99, phylum 99, class 98, and then it begins to drop. Like at species level, we only classify like 10% in this sample, but the others are 2%, 4%, 5%. So we can see that with green genes, which is a 16S database, at the species level, we get very low coverage. At genus level, we still have 20%, 50%, something like that. In change with the RefSec database, we can see that we have around 30 at the domain level. Yeah, at phylum level as well. And then when we go to species, we still have 20, 26, 13%. Like those numbers are much better at the species level when using the RefSec database. Um, yeah. And then we have yeah, some top 10 hits distributions per rank for all the samples. Like we can, for example, go to the family level and see that in this sample we have S24/7 in those quantities, And in the other ones, it's also the top identified family. Um, yeah, to compare the data. We can export this as a PDF. All those reports, they can always be exported as PDF to print them or to show them to a colleague or whatever. Yeah, so let's look at the statistics, the plots that we have. We can create rarefaction curves to judge our sequencing depths. We can select the level where we want to do this. Let's go for the genus level. Yeah, so we can see we go very steep here. So this is the number of distinct genus on the y-axis, and on the x-axis we have yeah, a random sampling of 1, 2, 3, 100,000, 200,000, 300,000 reads, and we always look at the distinct genus, and we want to have this very steep and then go to an asymptote. So here we have some that would benefit from more sequencing to find all the different taxonomies, but those are maybe quite well. They look not too bad. This is the whole genome shotgun sequencing example. Also looks quite well. Okay. Then we can look at the SHA-1 diversity curves. Again, we can select the different samples here we want to include and the level at which we want to do that. I already did that. And this looks very nice. Steep curve at the beginning and then asymptotic. The maximum hits stays on the same level. So this data set was sequenced with enough depth. We can say that. Okay, so then we have the numbers here and we have the rarefaction. That's fine. Let's look at the let's visualize the different taxa. So we can create a chronochart for that, pie chart. We can select the different samples we want to include. We combine the data in one chart here. So it looks like this. This is a very popular graphic, highly customizable and very interactive. So we can go into a certain taxa and then look at the numbers, and we can hide those small groups. We can increase the font size here, and we can look at another sample and see how it shifts. What are the changes? How do they differ? Okay, and you can also search for a specific taxa and it will move you there, and then yeah, so this is very handy for one by one sample comparison. We can also look at create a bar chart. Um, yeah, let's look at those. That looks a little bit better. Let's customize this a little bit. So those are two different ones. One is for the green genes dataset and the other one for the green genes database, and this one is for the I created this for the RefSec. You can see that we have a lot of unclassified here. Like those are the different samples and those are the percentages of taxonomies. There's bacteria here and a lot of unclassified. I can hide the unclassified and see that yeah, most of the classified are bacteria. I can go down to phylum and I see that those samples have a lot of Heumgongvirae, so that's some type of virus, I would say. And I can go further down to the family level, genus level, even species level, and I can see the different compositions. There are a lot of small bars here at the right, representing very little percentages of the found data. I can group them with a threshold. Everything that represents less than 1% will be grouped, and now it looks much nicer. Um, I can see, for example, that in this group here we have a lot of Alobaculum and Bifidobacterium, which is not so present in this part and in those samples. So I can visually compare um, everything, and we can play around with the visualization. I can extend the multi-line legend. I can increase font sizes and all that. So that's the stacked bar chart. You can see outliers very fast here, and yeah. So, but still, sometimes it's not that clear. And like here we can see those groups, so there's something going on, but sometimes it's not that clear, and we want to see how those samples separate. And for that, we can create a principal coordinate analysis plot. I did that for the RefSec at the genus level. And let's see. So you would go to charts, click the PCOA button, and then you can select the samples you want to include. You can provide an experimental design. If you don't provide an experimental design, you will get gray dots here, and that's it, and with the sample names and no more. But if you want to, you can add the experimental design. The experimental design, is a text file. I downloaded the spreadsheet from the study, and yeah, do a little bit of formatting, one sample per line, and then we have those headers where it says experiment and condition, and I put all those values. And when I load that into here, I can see all of them, and I can see the where they like what experiment each sample belongs to and the condition and everything. And I click next, and I can select the how I want to color the plot and taxonomic level and some stuff here to create the PCOA. Hitting the run button, I will get this graphic, and because I selected the experimental design, I get the colors. I can change the colors here. I can say, so first of all, this contains week 100 mice, week 20 mice, and then co-housing aged and co-housing young. And I can see that week 100 separates from the others. So the ones that are co-housed, they are much more similar to week 20 than to week 100. So there's definitely something going on with the co-housing. It has some effects at the taxonomic composition um, level. I can change colors. I can make this, for example, green, and now I cannot distinguish it anymore from the 20 week. Um, yeah, so this helps you to cluster stuff, to see how they separate, and yeah, to compare with your hypothesis. That's very handy. And once you are confident with that, you can go, or we can do a differential abundance analysis of the different taxonomies. So this is done with HR internally. We have some sample filters and counts per million filters here, normalization methods, and we can again provide an experimental design. If we don't have an experimental design, we can select the samples one by one, and we want to test against those samples here. But it's much better to provide an experimental design and then go for the factor you want to compare, like where you want to compare, in this case, conditions. And we want to compare, for example, week 100 versus week 20. And then hit the run button and have that comparison done. So I already did that. This is week 100 versus week 20. And this is co-aging versus week 20. So let's look a little bit at that. That's a typical, differential abundance chart. We can sort by tags, and we see one taxa per line. And we can see that some of them are underrepresented, some of them are overrepresented, and we see the fold change, the log fold change, log CPM, the p-value, the FDR, and we get a small report where everything is summarized, how many over, how many underrepresented, with which filter values, because we can adjust the filters. And we see the experimental design. And yeah, so as I said, we can adjust the filters here. We can say, those are so many taxa. I wouldn't know which one to look at. This is very general. I want to be more strict. I set the p-value filter for FDR to 0.005. And as you can see, I reduce the results. Like it's much less now. So those are much more like strong, no? The effect is much stronger on those taxa. And so I looked at those, and I can tell you that the ones that are more present in the younger groups, like in the 20-week samples or in the co-housing samples, they are mostly related to like those taxa are mostly related to reduce inflammation in the gut, and the ones that are more present in the aged mice they favor inflammation mostly. So yeah, it makes sense somehow. Um, but I will leave the interpretation at this point. I will leave that up to you. We can also create heat maps for those results. We can select the top 10, 30, 50 or species we are mostly interested in, and then some parameters here. So we get this kind of heat map where everything is separated. We can customize this. We have the experimental designer at the top, and then we have one species per line. You can see Akkermansia here, for example, which is more present in or like it's overrepresented in the week 20 group compared to the week 100 group. And it is related to rejuvenation effects. So we can customize this a lot. We can remove the dendrogram. We can remove the cell values, make this a real heat map, change the colors. and we can export this again, the data and also the PNG. So yeah, that's basically what I wanted to show you. So we have seen that we can do comprehensive metagenomics analysis with OmicsBox. There's a lot of different functionality that you can combine together. The tools and the databases are highly adaptable for different scenarios. And we have also seen that OmicsBox is a self-sufficient platform. You don't need to go outside. You don't need to know any bioinformatics. But if you want, you can always export the text data of all the analysis that we did in OmicsBox, and then you can go into a different tool, into R or into a spreadsheet program if you need that. So thank you for your time. Thank you for staying till the end. This concludes my presentation, and yeah, thank you again.